the whole purpose of this entire thing and why this is so difficult. And I will tell you that when I took microbiology, I never understood this when I was in college. And the reason is that I had to learn all the individual steps and the intermediate products and the enzymes involved. And there was just so much detail, like, I couldn't see the purpose through all that detail. Okay? So what my goal for you is to not have as much detail and look at the big picture and why do we care, right? And I will tell you that in my course, I really only limit your content to whatever you absolutely need to know for the rest of the semester to make sense. So even though this sounds kind of like basic biology and you're like, I can ignore this, the reality is that a lot of what we're going to talk about in the future is not going to make sense if you don't get this. So we need to make sure that we get it now. Okay? So the purpose of metabolism, as you hopefully know, is that microbes and actually all cells take larger molecules and break them down into smaller ones to generate energy in the form of ATP. Okay? And then they use that ATP to make larger molecules again. And we're going to talk about that in future chapters. So we use ATP to make peptidoglycan to build cell walls. We use ATP to replicate our DNA. We use ATP to make RNA. We use ATP to make proteins. We use ATP for making all the chemical reactions in the cell that cause microbes to grow. All of that costs ATP. And where's that ATP coming from? Okay. The thing to know about ATP, that ATP doesn't float around in the cell, kind of like, oh, I'm gonna just chill out here. As soon as ATP is made, it's almost immediately used for something else. There's never an excess of ATP in a cell. Okay. If there is ATP available, it will immediately be used for something. If there's a lot of ATP, then the cell will just divide and start growing and growing and growing like crazy because there is a lot of ATP to grow. But there is not going to be any ATP floating around. It's like, well, I don't know what I'm going to do today. That's not how it works. Okay. So the purpose here is that the cell gets ATP through catabolism, which is the breakdown of larger molecules into smaller ones. Now, there are a lot of different larger molecules that microbes can use for this, but ultimately, all of these molecules are broken down into some form of glucose or fructose, some kind of sugar, typically glucose. And this is why we start our journey here with one molecule of glucose. And we're going to see what happens to one molecule of glucose, and then you need to realize a cell has millions of molecules of glucose. So this is going on at a much larger scale than what we're talking about here. Okay? And there are some exceptions to this, but for purposes of today's class, all microbes start their catabolism with a molecule of glucose and a process called glycolysis. And glycolysis literally means sugar breakdown, like glycolysis, sugar, and breakdown. And that's exactly what it's going to do. It's going to take one molecule of glucose and break it down. Now, glucose is a very energy-rich molecule. By breaking that down, the cell will be able to harvest that energy and make ATP. And that ATP will be used for these other processes in the cell. So let's start with one molecule of glucose. And I will mention it here. You don't have to know the structure of glucose, but if you've ever seen it, it may make sense what I'm talking about. But glucose is a molecule that has 6 carbon atoms. Okay, it also has 6 oxygen atoms and 12 hydrogen atoms. But for purposes of this discussion, that's not as interesting. We're going to follow what happens to the carbon atoms. Okay. And glucose is going to undergo glycolysis, which literally means sugar breakdown, right? And glycolysis consists of 10 individual biochemical reactions. And I don't need you to know any of them. We're going to pretend that glycolysis is one reaction, one and done. Okay? The reality is a little different. Now, glucose is a very energy-rich molecule, but it's also very stable, chemically very stable. It's hard to break down. So we actually need to use a little bit of energy to break it down. One way to think about this is maybe like there's a glucose molecule and we used 2 molecules of ATP. We take those phosphate groups and stick them onto the glucose molecule, kind of like flags. And then adding that phosphate to the glucose makes the glucose unstable and it basically falls apart. Okay? But in short, 2 molecules of ATP are necessary to break down 1 molecule of glucose. Okay, so we're going to actually invest some energy. We also need 2 molecules of this compound called NAD, which is a, or NAD+, which is an electron carrier. NAD is the oxidized version. When we add electrons to it, it becomes NADH. And then NADH is the reduced version. If we rip electrons from NADH, it becomes oxidized to NAD+. So it goes in a cycle. NAD plus electrons makes NADH. NADH minus electrons becomes NAD+. So far? Okay. Now, what happens? What do we get out of this glycolysis? So out of this, we get 2 molecules of pyruvate, or pyruvic acid, depending on which textbook you read, right? And we make 4 molecules of ATP, right? And we make 2 molecules of NADH. Okay. Now we invested 2 ATP, we got 4 ATP out of it, so our overall yield of ATP is going to be 2 ATP. And actually what I'm going to do, I'm going to put this in a different color here. Hang on. Okay. Yeah. So I'll put whatever. Oh, annoying. Here we go. Okay. I'm going to put the yield in blue. So we're going to get 2 pyruvate plus 2 molecules overall of ATP plus 2 molecules of NADH. So far, so good? Okay. And so I'm interested. Yes. And pyruvate is a molecule that has 3 carbon atoms. So you see what we did? We took that glucose and we basically chopped that baby in half, right? Because the glucose had 6. And we chopped it effectively in half and made 2 molecules of pyruvate, each with 3 carbon atoms. Yeah, right. Right? So we broke down glucose in a process called glycolysis. And basically what we do is we take that 6 carbon molecule and go, whoop. It's a bit more involved than that, right? There's 10 different biochemical reactions, but that is effectively what happens in those 10 reactions, okay? And although we invested 2 ATP and we got 4 ATP out of it, our overall yield, if I ask you, well, I started with 1 molecule of glucose, what do I have at the end? I have 2 more ATP than when I started, right? And that's why I'm saying here there's 2 ATP. Okay. All cells start their glucose catabolism like this, no matter what they end up doing afterward. There are 3 major metabolic pathways that are possible, but no matter which one they're going to end up doing, they all start here. Okay? And then this is basically decision point. Now that we've undergone glycolysis, we have 2 pyruvate. The question is, now what do we do? And there are basically 2 options at this point. Okay? Cells can either go the route of fermentation or they can go the route of respiration. Okay? In fermentation, those 2 pyruvate that we got out of glycolysis, they are converted, these guys are converted to either acids or alcohols, sometimes also with carbon dioxide gas produced. Okay. And what happens is those 2 pyruvate then end up in 2 acids or 2 alcohols. And interestingly, we reduce the 2 NADH is going to be put back into, made into NAD+. So let me take this NADH here. It's also going to go, right? So we take the end products of glycolysis, the pyruvate and the NADH. The pyruvate is going to be converted to either acids and alcohols. The NADH is going to be cycled back to NAD+. Okay? The NAD+ now can, go back to glycolysis and do that whole thing again. See that? Right, because we need it. Right, because we need it. Yeah, so we started with a molecule of glucose, and we needed NAD+ to do this, right? And a little bit of ATP. And then when we're done, we get 2 pyruvate, which is converted to acids and alcohols, and the NADH is converted back to NAD+. This NAD+ can now go back to glycolysis and redo this. And so as long as we have glucose available, this can go over and over and over. Right? A cell has a very limited amount of NAD+ in the cell. So if we were to do glycolysis without the fermentation step, if we only would do this part over and over and over, eventually we would run out of NAD+. Like we wouldn't run out of ATP because we're actually making more ATP than we started with, right? So that's not a problem. But I would run out of NAD+. And then if I don't have any NAD+, no matter how much Right. Right. Even if I had billions of glucose molecules in the cell, if I don't have NAD+, it's just going to sit there because I need it to run glycolysis. So interestingly, this fermentation step, I don't know if you noticed here, but I took pyruvate, converted it to acids and alcohols. I took NADH and converted it to NAD+, but I didn't make any additional ATP. Right? No extra ATP. The only purpose of fermentation from the cell's point of view is to regenerate NAD+, so that glycolysis can continue. So cells that use fermentation, and there are microbes, there are bacteria that only use fermentation for their energy production. The only ATP they generate is out of glycolysis. That's it. Nothing else. For every molecule of glucose, they get 2 ATP, end of story. Okay, so fermentation is easy. So I guess I'm not a fan of how you phrased it. So the breakdown of one glucose molecule yields 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. If a cell goes through fermentation, then the pyruvate is converted to either acids or alcohols, the NADH is recycled back to NAD+, and the ATP goes other stuff. It goes for like making more DNA or making peptidoglycan, you know, all the things that a cell needs energy for. Okay? But the actual fermentation step, in the step from pyruvate to acids and alcohols, no additional ATP is made. This is often a trick question that my students miss on the exam. I will ask you this, like how much ATP is made in an organism that ferments sugars after glycolysis? And the answer is zero, nada. Okay? Okay. So now other organisms, instead of fermentation, they go the route of respiration. And whether or not they go one way or the other depends on what kind of organism it is. Like some organisms cannot do respiration, is not even an option, right? Other organisms can do one or the other, and then it depends. Like if they can do respiration, they'll do respiration, because the respiration will generate more ATP. But sometimes they can't. And we'll talk about why not later. And if they can't, then they do fermentation. Okay, and for example, the yeast that you're growing in your house right now for streaking for isolation, it can do either aerobic respiration or fermentation. But for aerobic respiration, it needs oxygen. If there is no oxygen available, it's going to switch to fermentation because it can happen without oxygen and still generate a little bit of ATP. Okay, like for example, when we make bread, we cover it up with like a towel and all that. What we're doing is we're limiting the amount of oxygen that we're allowed to get through. The yeast is going to take the glucose that we're providing it and make it into alcohol and carbon dioxide. And the carbon dioxide is what's going to make holes in our bread. And because carbon dioxide gas is going to make the holes. And actually, when you're fermenting your bread, when your bread rises, it actually makes alcohol. And of course, then you stick it in the oven and you burn off some perfectly good ethanol. So you don't actually have alcohol in your bread as a result, sadly. Okay, but organisms that have a choice will go through the route of respiration. So what is going to happen now, instead of going through fermentation, the pyruvate is going to go do something else. It's going to go ultimately to the Krebs cycle. But pyruvate cannot enter the Krebs cycle directly. It needs to undergo a sort of intermediate step. Depending on the source that you're reading, sometimes the next step is sort of considered part of the Krebs cycle, but it really isn't. It's really a separate sort of preparatory step for the Krebs cycle. And this is called the decarboxylation of pyruvate. And why they give these things difficult names, don't ask me. Nobody ever asks me for my opinion when they name these things. But you can figure out what it means, right? D means to remove. Carbo refers to a carbon atom. So really what it means is we're going to remove a carbon from pyruvate. Decarboxylation of pyruvate. Pyruvate has 3 carbons. What's going to happen if we remove one? How many are going to be left? So if pyruvate has 3 carbons and I remove one, yeah, right? That's what's going to happen too. So we start out with 2 molecules of pyruvate, right? They're coming from there. Because we didn't go through fermentation, we're going to go this other step. So we still have our pyruvate left, right? And we're going to remove a carbon, and that the carbon is going to be removed in the form of carbon dioxide. And because we have 2 pyruvate, there's going to be 2 carbon dioxides, right? And here, this is the carbon that we're ripping off of the pyruvate. And we're going to have left acetic acid, really, which is 2 carbons. And what we do at this point is we actually attach it to a handler. And the handler is called coenzyme A or CoA. And so the final result is going to be 2 molecules of acetyl-CoA. Okay, I never understood this when I was in class. I was like, what the heck is that CoA thing and why do I care? And the answer is you don't have to care. The coenzyme A is basically a grabby thing that sort of holds on to the 2 carbons that are left over and brings them to the Krebs cycle. And other than that, the CoA part doesn't play a role. The important component here is the acetyl part. This thing here has 2 of those carbons. So this is what happened. We took 1 pyruvate, ripped off a carbon in the form of carbon dioxide, and the 2 other carbons are in the acetyl-CoA, right? And we did that twice because we had 2 molecules of pyruvate, so everything is doubled. So actually what I want to do is I want to put this in blue as well as my end product here. Hang on. Okay, so I'm going to have 2 acetyl-CoA. And the acetyl-CoA is what's going to move into the Krebs cycle. Okay, now, kind of like glycolysis, the Krebs cycle is this. I'm sorry, what did you? Yes, right. And the Krebs cycle is this complicated pathway with many different steps, and you don't have to remember a single one of them. You just need to know there's lots of steps. Okay, acetyl-CoA is being fed in here. You do need to know what comes out of it. So you need to know what goes in, what comes out. Acetyl-CoA goes in, and what comes out? For each acetyl-CoA, we're gonna get two carbons out of it. And we had 2 acetyl-CoA, so we're going to have 2 times 2 equals 4 carbons. And they're all going to come out in the form of carbon dioxide. So we started out with 6 carbons in glucose. 2 of them here were removed in the form of carbon dioxide, and the other 4 are removed in the form of carbon dioxide in the Krebs cycle. So by the time we're done, we have completely oxidized glucose, like all those 6 carbons that we started out with have been removed in the form of carbon dioxide. All of them are gone. Okay? So the other thing we do is we make 2 extra molecules of ATP. So that's nice, right? We make a little bit of ATP. And then we make a little more of that NADH. Oh, actually, I forgot to mention here. Oops, hang on, back up here. In the decarboxylation of pyruvate, we also make another 2 molecules of NADH. This is going to become important later on. So I don't want to leave it out. Okay, and then in this step, in the Krebs cycle, we make 6 more molecules of NADH. And we make another electron carrier called FADH2, okay, which does the same cycling thing. So you have FAD, which is the oxidized version, takes on electrons, they become FADH2. And then when you take FADH2 and you rip off these electrons, it converts back to FAD. So it goes in a cycle, FAD, FADH2, FAD, FADH2. Okay, so in this case, we're taking yes, you are right, my friend. 2. Okay. Okay? So whenever we break glucose down into these little pieces, we get energy becomes available. Some of that energy is used to make ATP, some of the energy is used to make NADH, and some of the energy is going to be used to make FADH2 in the Krebs cycle. Okay, so that's nice. Now I have NADH, I have FADH2. Oh, do you see that just by going this step now, I have made 2 additional ATP. To be perfectly technically honest, it's actually GTP that is being made, but for purposes of our class, I don't care. If you say ATP, GTP, either one is fine, okay? So you don't have to remember something different for here. By going this step, now we've made 2 more molecules of ATP. And so now we have a total of 4 ATP. We made 2 in glycolysis and we made 2 more in the Krebs cycle. So by going this way, we've already doubled the amount of ATP that we get out of the same molecule of glucose. Right? So can you see that if an organism has a choice, it'll go the route of respiration rather than fermentation? Because from 1 molecule of glucose, I can get 2 molecules through fermentation of ATP. And in respiration, I can get 4 molecules of ATP. But wait, there's more. But are we good so far? Okay, I have to throw a little more terminology at you. The mechanism that we use to make ATP in glycolysis and in the Krebs cycle is called substrate-level phosphorylation. Okay. That means phosphorylation means to add a phosphate to something. So we're taking ADP, right? And then we're adding an inorganic phosphate to it, and that results in ATP. Because this is, I don't know if you know, you don't have to know this abbreviation, but ADP stands for adenosine diphosphate, which is adenosine with 2 phosphates. And ATP is adenosine triphosphate, which means adenosine with 3 phosphates. So we basically add another phosphate to it here. Okay, and this step here, this is called phosphorylation. And substrate-level phosphorylation is the mechanism by which we make ATP in glycolysis and the Krebs cycle. Just as part of that biochemical reaction, we take ADP, we get some energy, we use that energy to take inorganic phosphates, squish them together, and voila, ATP is made. Okay, that's going to be a slightly different mechanism that we're going to see later in Aaron's favorite electron transport chain. Okay? Are we good so far? So we made 4 molecules of ATP, which is nice. And then we made a whole bunch of that NADH and the FADH2. And you wonder what the heck is going on and why do I care and why do I need this? Let me save this picture because we're gonna run out of space here. So I kind of want to start a new sheet here. Yeah. But it's kind of important that we come back. Okay, I'm gonna try and do this neatly, although I am not the neatest drawer, but I will try and draw it small and keep it on the same page, but bear with me. Okay, so this all happens in the cytoplasm of the cell, okay? And this is because bacteria do not have mitochondria. In eukaryotic cells, this happens in mitochondria, although the glycolysis happens in the cytoplasm. But we're going to focus on bacteria. Okay, so all of this happens in the cytoplasm. So imagine that, this is the cell membrane. This is the inside of the cell. And this is the outside of the cell. Okay, and for purposes of this discussion, we're ignoring that there may be a cell wall on the outside and all that good stuff. We have enough going on as it is. Okay, in the cell membrane or plasma membrane of the cell, there are a whole bunch of molecules, and these molecules here are in a specific order, and this is called the electron transport chain. Okay, and they are all ordered in a very specific order from the organism, from the molecule that in order of their ability to accept electrons. Like the further down you go, like the further you go that way, the better able they are to accept electrons. Okay? Okay. So, what is going to happen? So, we're going to now focus on one molecule of NADH. What happens to that molecule of NADH? NADH that we made, and we made this either in glycolysis, or we made it in the decarboxylation of pyruvate, or we made it in the Krebs cycle. We made a whole bunch of NADH, right? And so what's going to happen is NADH is going to travel to the cell membrane, like right over here, right? And it's going to donate those electrons that it picked up earlier. And when it does that, it is converted back to NAD+. This NAD+ here can go back to either glycolysis or the decarboxylation of pyruvate or the Krebs cycle and go pick up more electrons there, being converted back to NADH. Then the NADH travels through the cell membrane, donates those electrons, becomes NAD+. NAD+ goes back again to those earlier steps. Okay? So it's a kind of a complicated cycle, but that's what happens. The NAD+ picks up electrons here or here or here, converts to NADH. NADH then travels to the membrane and donates those electrons there. And then NAD+ is free to go pick up more. Okay. Now, these electrons are going to be passed down through the electron transport chain. I mean, it's the chain that transports electrons, right? So the electrons are going to come from here, or going to go here, and then here, and then here, and then here. And then eventually they're going to end up on what we call the final electron acceptor. Okay, and at this point, I'm not going to name the final electron acceptor. We're going to leave it blank for now. But there is a molecule somewhere inside the cell, in the cytoplasm, that's going to accept those electrons after they have traveled through those proteins. Now, what do you think of when you think of a flow of electrons? What is that? Or do you think of anything? Maybe not. I guess I'm a nerd. Can you think of flowing electrons as a source of energy? Like in the real world, where do electrons flow? You don't, are you familiar with the concept of electricity? Do you know what electricity is? Yeah, so that is actually, electricity is the flow of electrons. Electrons flow through wires, and that's what powers your life, your cell phone, your fridge, your everything. Everything runs on electricity. And electricity is a flow of electrons. There's a current of electrons that runs through wires, okay? So the flow of electrons through these electron transport chain is a form of energy. It's a form of electrical energy. Right? And that energy comes ultimately from NADH that donates those electrons. Okay. So what is going to happen with all that energy? This protein, the first one in that electron transport chain, is going to use some of that electrical energy to pump hydrogen to the outside of the cell, or protons. These are H plus atoms, right? H plus atoms are known as hydrogen atoms or protons. And we'll figure out why that's important. Okay. Not all the proteins in the electron transport chain can do this, but some of them do. Like this guy does it. It will take the flow of electrons and use that energy to pump hydrogen to the outside. Kind of like that. Okay, and then this last guy does the same thing. And you don't need to know the names of these proteins. Okay, I'm trying to really focus on the big picture here. So as these electrons are flowing through the proteins, those proteins are using that flow of electrons as energy to pump hydrogen to the outside. Now, the other thing we need to know about electrons is that when molecules take on electrons, they become reduced. When they lose the electrons, they become oxidized. So each of these molecules is undergoing a reduction and then an oxidation, and a reduction and then an oxidation, right? This guy, so NADH is reduced. It gives off the electrons to this protein here. NADH becomes oxidized to NAD+. This protein here becomes reduced when it takes on the electron. When it passes the electron on, it becomes oxidized again. Then the second molecule becomes reduced, passes down the electron, becomes oxidized. So each of these goes reduced, oxidized, reduced, oxidized, reduced, oxidized, reduced, oxidized, and so on and so forth, right? Until the final electron acceptor lands on another molecule that becomes reduced, and that is the end of the chain. Okay. So now what? So now the NADH has lost all its energy. It's donated the electrons here. We use that energy to pump protons to the outside. And now we're going to actually use those protons to make ATP. Okay? It seems like a very convoluted way to make ATP, but you'll see in a minute why we want to do this, if you just bear with me. So can you imagine that all these protons are going to be pumped to the outside, and really what you're going to get is what we call a concentration gradient. A lot of protons on the outside, very few protons on the inside, right? You also get a charge gradient, highly positively charged on the outside, relatively negatively charged on the inside. And you get what we call a pH gradient. If you've ever taken chemistry, you know that high amounts of protons in a solution actually makes something acidic, so this becomes more acidic and the inside becomes more basic. Okay? So really what would, and nature abhors this sort of imbalance. And the hydrogens really would like to flow back into the cell and sort of make the charge and the concentration across that membrane equal. But the problem is the cell membrane is made out of these phospholipids. Let's put them in here just for a sec here. And I'm going to put them in here so they don't mess up with my drawing, right? The cell membrane is made out of these phospholipids with these charged heads, right? And very hydrophobic chains in the center. And anything that is highly charged, such as my protons, will not be able to pass through the membrane. Because as soon as they try, the hydrophobic lipids will push them back out. It's like, you know, what the heck, you don't belong here. So I imagine, so here's me with my imagination, and as you can see, I have quite a bit. But I imagine these protons on the outside of the cell sort of bumping up against the membrane, like, let me in, let me in, let me in, right? And that pesky membrane won't let them in because it's too hydrophobic. Along comes a protein to the rescue called the F1 Atpase. The F1 Atpase is a molecule that runs, it's actually a very large molecule. Okay. This is the F1 Atpase. The F1 Atpase or F1 of ATP or Atp Synthase allows protons to flow back into the cell. And you want to go back in? Okay, I'll let you back in. But we're going to go for a trip. So there is a channel here in the ATPase, this large enzyme that allows the protons to go in, but instead of letting them straight in, it actually takes them for a little trip around the protein, kind of like that. Like here's my channel, right? And then it allows the hydrogens to move back to the inside. Yay. Things are back to normal. Okay. Now that ATPase uses the flow of those protons, and you don't have to know how it works, but really what happens is there are these subunits with alternating negative and positive charges that sort of attract and repel, are attracted and repelled by the flowing protons. So the highly positively charged protons are going through that protein, and that protein in return will turn. It will run in circles, kind of like a little engine. Okay. Yes, that's how the protons get back in, right? And so the protons get back in by going in a circle around the protein, allowing this protein to rotate in a circle, kind of like a propeller. Okay. That propeller movement is actually conferred onto the stalk here. There's a stalk. Let me make the stalk a different color here. This starts turning as well. And that movement changes the shape of this part here, what we call the lollipop head. Because it looks a little bit like a lollipop. Okay. The lollipop head takes that movement and it changes shape, and the lollipop head can take on 3 different positions. One position in which it's open and can bind ADP and inorganic phosphate. And then as the stalk turns, it squishes them together to make ATP. And then the stalk turns again, opening it up and releasing ATP into the environment, into the cell. In the meantime, more protons are going around, and this little machine here keeps running and running and running and running and running and changing the shape of the lollipop head to bind ADP and inorganic phosphate, make ATP, squish them together, open it up. Bind, open, bind, open, bind, open. And that's how ATP is made. Okay, so I'm going to backtrack a bit. So ATP is made by the shape changes of the lollipop head. The energy from those shape changes comes from the movement of the stalk. The movement of the stalk comes from the flow of protons to this ATPase. The protons were pumped to the outside by the electron transport chain, right? The electron transport chain got that energy from the flow of electrons. The electrons were coming from NADH. And NADH was coming from the Krebs cycle, from the decarboxylation of pyruvate, and from glycolysis. It's a long way to go. But for every molecule of NADH that donates electrons that way, I can get 3 molecules of ATP through this mechanism, which is called oxidative phosphorylation. And it's called oxidative phosphorylation because of all the reduction-oxidation reactions, right? That happens. Oxidative phosphorylation. Okay. How many NADHs? Actually, I need to put that NADH there. How many NADHs did I make from 1 molecule of glucose in total? How many did I make here? 2. How many did I make here? How many did I make here? So how many did I make in total? 10. How much ATP is that going to give me ultimately? 30 ATP. Like, holy moly. Can you remember the times that we were happy that we made 2 ATP in glycolysis? And that we were like, oh my goodness, you can go through the Krebs cycle and you get 2 more ATP. Yay, 4 ATP. I can pull like 10 NADHs out of this. I make 30 ATP. But wait, there's more. FADH2 does the exact same thing as NADH. It donates electrons to the electron transport chain, and protons are being pumped to the outside, and ATP is made, yada, yada, yada. The only difference is that FADH2 is not quite as good an electron donor as NADH is. So it cannot donate those electrons at the very first protein in the electron transport chain. It donates these electrons somewhere over here. So FADH2 donates electrons there, reverts back to FAD, right? And then the FAD can go back to the Krebs cycle, pick up more electrons, and return to FADH2. FADH2 donates electrons here, and then from here they go the same way. They go like there, boing, 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 and boing, right? But because it starts later in the chain, it doesn't use that very beginning of the chain. It doesn't use this part, right? It starts here and then it goes in that direction. Because it goes in order of the better electron acceptors, it can't go backward, right? Exactly. It starts a little later, and therefore FADH2 doesn't result in as many protons being pumped to the outside because it misses out on this first step. It starts here, and these protons are still being pumped to the outside, but not those. So as a result, for every FADH2 that donates electrons here, I can get 2 ATP and not 3. And I made 2 FADH2 in the Krebs cycle, right? So they give me a total of 4 ATP. Right? Okay. So this all assumes that we use along with what? Yes. 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 Right. Yes, right. So both NADH and FADH2 donate these electrons as Right. As long as electrons are being donated, those electrons are going to flow. Those little proteins here are going to pump protons to the outside. Those are going to drive that little ATPase engine and make ATP. Yes, this is going to go on all the time. Okay. Now, this all assumes that way more, right? You see, it's a pain in the butt. Sorry. Excuse my French. It is hard to go through all these chemical reactions, but isn't it worth it if you can make so much ATP? Right? Okay. Now, this assumes that we're using the entire electron transport chain and not just part of it. And when can we use the entire electron transport chain? We use that if the final electron, it depends on how strong the final electron acceptor is. If we use a really strong final electron acceptor, then we can pick up electrons here at the very end. But if your electron acceptor is meh or not as good, then it will pick up electrons maybe here or here, right? And so then you don't get as much ATP out of this. So how much So how much ATP you ultimately get out of this depends on which final electron acceptor you use, and therefore where the electrons end up in the final step. Okay. The final electron acceptor that you've probably heard about is, which one have you ever heard about? Oxygen, right? So oxygen, or O2, molecular oxygen, is a common final electron acceptor, and oxygen is the best electron acceptor in nature, in biological systems. Okay? And so when oxygen takes up this electron, it becomes reduced and it turns into water, and then water is your waste. And then when we use oxygen as a final electron acceptor, we call this aerobic respiration, because aerobic means with oxygen. So aerobic respiration means we use oxygen as a final electron acceptor. And if we use oxygen as a final electron acceptor, then we use the entire electron transport chain. And so then we get a total of, let's look at total ATP yield. I'm running out of space on my thingy here. So we use aerobic respiration. We get 2 ATP out of glycolysis, 2 ATP out of the Krebs cycle, 30 ATP from NADH through oxidative phosphorylation, and 4 ATP from FADH2 through oxidative phosphorylation, for a grand total of 38 ATP. Holy moly. For that same molecule of glucose that we started out with. One molecule of glucose gives you 38 ATP if you go through aerobic respiration. That means you start with a molecule of glucose, go through glycolysis, go to the decarboxylation of pyruvate, go to the Krebs cycle, all the NADHs go to the electron transport chain, all the FADHs you make go to the electron transport chain, you run your ATPase enzyme and you make a whole bunch of ATP. Now, some organisms do not use oxygen as their final electron acceptor. In fact, they may not even have this last enzyme. Like, they may not even have it, right? And so they cannot possibly use oxygen if their life depended on it. And in fact, their life does depend on it. So in most cases, those organisms, instead of using oxygen, they use sulfur, like S2. And that becomes reduced to hydrogen sulfide, which is a gas, a stinky gas. It smells like sewage or rotten eggs. Have you ever smelled sewage? Okay, what you're smelling is the hydrogen sulfide that is produced through anaerobic respiration by the bacteria in that sewage. So microbes that use any other electron acceptor here, like sulfur. Sometimes you can use nitrate or nitrite or all sorts of even organic molecules, like different organisms do different things. They almost always make stinky gases, and that's what you're smelling. In fact, you have anaerobic bacteria in your gut and you smell their results every single day. Right? The stinky gases they make, you smell them too. Okay? Those are all the result of anaerobic respiration. And because sulfur is not as good an electron acceptor as oxygen, it cannot pick up the electrons at the end. It picks them up maybe here or here, right? And therefore, you don't use the entire electron transport chain. You cut off the end. You use a shorter one. And therefore, you don't get quite as much hydrogen being pumped to the outside. Therefore, you get less hydrogen to the outside, less ATP is being made. And therefore, anaerobic respiration always makes less ATP per molecule of glucose than aerobic respiration does. How much depends on which finite electron acceptor it uses. You don't have to know that, as long as you know that it's less than 38 ATP, but it's more than 4. Because just by going through glycolysis and the Krebs cycle, we've already made 4 ATP. Okay. Okay. Yes. Yes. Makes 30. One NADH results in the production of 3 ATP, one FADH2 in the production of 2 ATP under ideal circumstances, assuming that we're using oxygen as a final electron acceptor and we can use the entire electron transport chain. If we don't, it'll be less. And then it depends on which one we use. So let me make another little separate box here for you. As you can see, I am no artist, but... So let's put all the possibilities here. So if an organism starts with a molecule of glucose and goes through fermentation, it makes a maximum of 2 ATP, the 2 ATP that comes out of glycolysis and nothing else. If an organism goes through aerobic respiration, it will make a total of 38 ATP per molecule of glucose. There you go. If we use anaerobic respiration, that means we use a different final electron acceptor. We make more than 4, but less than 38 ATP. So intermediate. Individual organisms can do individual things. So some organisms can only use aerobic respiration. If there's no oxygen around, they either die or they make endospores to sort of survive. Right? Some organisms can only ferment. Other organisms can choose, like yeast, can you choose between aerobic respiration or fermentation. And some organisms can choose any of them. Like my friend Salmonella, if there's oxygen around, it'll use aerobic respiration. If there's no oxygen around, but there is sulfur, it'll use anaerobic respiration. If there's no oxygen or sulfur around, it can ferment any number of sugars and just go through fermentation. Like it's the master of survival. It'll eat anything. Okay. But then Pseudomonas aeruginosa is a gram-negative bacterium. It can only, it's obligate, what you call an obligate aerobe, you'll learn that term next week, can only use aerobic respiration. I know this is a lot. I hope, Aaron, that I solved your problem with that electron transport chain and that you love it again. Yeah. Okay. Okay. Do you get it now? Okay. So let this sink. So give me some time. I have to be in class myself. My class started at 7. So I have to cut this short. But what I'm going to do afterward, I'm going to, no, not a problem. This is fine. I'm happy to do this. I will edit the video and post it in the Unit 1 forum. And so you can, try and figure out what I did here on your own again if you want to. And then if you have any additional questions, just put them in the Unit forum and I'll answer. Okay? You're very welcome.